Carbohydrates, Chapter 24. Objectives. 1. Introduction to carbohydrates. 2. Monosaccharides. 3. Disaccharides. 4. Polysaccharides. 5. Amino sugars. 6. Review of key reactions. Your goals. By the end of the chapter, you should be able to know sources and functions of carbohydrates, name and draw monosaccharides, predict reactions of monosaccharides, understand disaccharide and polysaccharide formation, understand structure of DNA and RNA. 1. Introduction to carbohydrates. It might be necessary to review the following topics to prepare for the material in this chapter. 1. Drawing chair conformations. 2. Haworth projections. 3. Fisher projections. 4. Hemiacetals and acetals. And 5. Reduction of aldehydes and ketones. Carbohydrates defined. Carbohydrates are also sugars, and they are polyhydroxyaldehydes or ketones, naturally synthesized in nature from carbon dioxide and water through the process of photosynthesis, and they function as an energy source, fiber, etc. 2. Monosaccharides. Monosaccharides are simple sugars. They can contain multiple chiral centers. Fisher projections are often used to represent their structure. Here are two examples of monosaccharides, glucose and fructose. Basic naming. An aldopentose and a ketohexose examples are shown here. Aldose contains an aldehyde group. The ALDO prefix is used. In a ketose, it contains a ketone group. The prefix keto is used. The terms di, tri, tret, pent, hex, hept indicate the number of carbons in that given monosaccharide. D and L monosaccharides. Consider glyceraldehyde, the simplest aldose. It contains one chiral center. D sugars have the OH group at the chiral center farthest from the carbonyl on the right-hand side in the Fisher projection, while L sugars have it on the left. So we want to find the OH group at the furthest chiral center away from the carbonyl carbon. If it is on the right, it's the D sugar. If it's on the left, the OH group, it's an L sugar. D sugars means configuration of the furthest chiral center is R, while the L sugar has the S configuration at the furthest chiral center. D ribose has that R configuration at the very last chiral center, furthest away from the carbonyl carbon. D-glucose, the same thing, and D-fructose. Note, dextrorotatory with a positive sign versus levorotatory with a negative sign does not correspond to the D and L designations that we use for monosaccharides. Aldotetroses. There are 4 possible aldotetroses. 2 are D sugars and the other 2 are the enantiomeric L sugars. The enantiomers of the compounds shown are called L-erythros and L-threose. Aldopentoses. Aldopentoses have 3 chiral centers. The number of isomers will then be 2 to the third, which means 8 possible stereoisomers. The D sugars are all naturally occurring. They include D-ribose, D-arabinose, D-xylose, and D-lysose. Aldohexoses have 4 chiral centers, so 2 to the 4 means that there are 16 possible stereoisomers. The 8 naturally occurring D sugars are shown here below. Overview. Here is a flowchart of some aldoses, starting with 1 chiral center, then continuing on to 2, to 3, and then 4. Ketoses. This is a list of some naturally occurring deketoses. Ketoses are polyhydroxy ketones. Cyclic structures. An aldehyde can react with an alcohol to produce a hemiacetal under protic conditions. Hemiacetal formation is favored for 5- and 6-membered rings. Hemiacetal formation. Monosaccharides undergo ring-closing hemiacetal formation, just like we saw previously. The new chiral center formed at the anomeric carbon is C1. The equilibrium greatly favors the cyclic form. 6-membered hemiacetal ring of a saccharide is called a pyranose ring. The stereoisomers formed upon cyclization are called anomers. Alpha anomer. That's going to be the anomer where the OH group is trans to the CH2OH group. Notice here that we have the CH2OH group on top and down here at the bottom. That's the trans species. If they're on the same side of the ring, that's the cis species. The beta anomer, this one's the cis species. Mutarotation. Mutarotation is an interconversion of alpha and beta anomers. The dynamic equilibrium between the alpha and beta anomers is called mutarotation. Mutarotation is accelerated with acid or base catalysis. With some sugars, the alpha form will dominate, but the beta anomer dominates for others, and it's hard to tell. Pyranose forms. A pyranose ring is either drawn as a Haworth projection or a chair conformation. Chair is better because it does reveal that 3D conformation. D-glucose is the only sugar where all groups are equatorial, and therefore it's the most stable sugar, thus the most abundant in nature. Ring oxygen always is drawn as the upper right rear position. So notice it's right here. Furanose forms. Furanose rings are 5-member rings of a hemiacetal of a sugar. Fructose equilibrates between both furanose and pyranose forms. The furanose form of D-fructose takes part in most biochemical reactions. Ester and ether formation reactions. Hydroxy groups of a monosaccharide can be converted to esters to improve their solubility in organic solvents. Excess reagent allows for a reaction of all free hydroxy groups. The hydroxy groups can also be converted to ethers via the Williamson ether synthesis. Silver oxide is used as the base. Glycosides. Cyclic hemiacetals of a sugar can react with an alcohol under acidic conditions to form an acetal. Acetals of sugars are called glycosides. Mixture of alpha and beta glycosides is obtained. Here is an example of the alpha, and here's an example of the beta. Mechanism. The mechanism of glycoside formation is analogous to the formation of a typical acetal. In the first step, the oxygen is protonated, which makes it into a good leaving group. Water leaves, forming a carbocation that is resonance stabilized. Now let's consider what occurs for the formation of the alpha glycoside. Once we have the carbocation that's resonance stabilized, an alcohol can come and attack the carbon. The pi electrons are bumped up to the oxygen. We have oxonium that is deprotonated by water, and then now we have the alpha glycoside where we have the groups on opposite sides of the ring. Formation of the beta glycoside follows pretty much the same steps. We have attack of the alcohol's oxygen onto the carbocation center. Then water removes H+, resulting on the beta glycoside where we have the groups on the same side. Epimerization. Under strongly basic conditions, Aldoses undergo epimerization at carbon 2 via the ene-diol, that is, enol intermediate. Epimers are diastereomers that differ in configurations at only one chiral center. Reduction. Monosaccharides can be reduced to alditols with sodium borohydride. D-glucatol, called D-sorbitol, is often used as a sugar substitute. Carbohydrates may also undergo oxidation. Aldehyde oxidation of an aldose produces an aldonic acid. A variety of oxidants can be used, but a mild oxidizing agent must be used in order to avoid oxidation of the alcohol groups. A stronger oxidant, like nitric acid, will oxidize the aldehyde and a primary alcohol of an aldose, forming an aldaric acid. Monosaccharide tests. Selective oxidation of an aldose and not a ketose is used as a chemical test to differentiate between the two. Oxidation tests also performed to detect the presence of reducing sugars. Pyranose forms of the sugar include Tollens test, which the test tube here would show a positive test, and Felling's test. Glycosides, that is acetals, are not reducing sugars and are not oxidized. Cyanohydrin. Recall cyanohydrin formation. It works on aldoses. A mixture of stereoisomers would be obtained. Kiliani-Fischer synthesis. The cyanohydrin is reduced to an aldehyde. Overall, the synthesis lengthens the carbon chain of the sugar by 1 carbon. The cyano group can be converted to an aldehyde in one step. For example, if we have D-arabinose reacted with first HCN followed by hydrogen gas in the presence of palladium barium sulfate and water, that will result in the addition of 1 carbon. Wool degradation. The wool degradation achieves the opposite of the Kiliani-Fischer synthesis. It shortens the carbon chain by 1 carbon. The reagents here given in this example with D-glucose as our starting sugar is NH2OH, which will form an oxime, followed by AC2O, which will result in a cyanohydrin, and then a strong base will give us D-arabinose. 3. Disaccharides. Disaccharides consist of 2 sugars connected through a glycosidic linkage. Glycosidic oxygen connects C1 to C4, so this example is called a 1,4-disaccharide. Maltose is a reducing sugar, in this example here. Stereochemistry. The linkage gives rise to stereoisomers. The pyranoses cellubiose and maltose only differ by the stereochemistry at the anomeric carbons. This one is the beta-glycoside, and this one is the alpha-glycoside. Disaccharide examples. This is an example of a disaccharide, lactose, which is also known as the milk sugar. Sucrose. Sucrose is known as table sugar. Honeybees can convert sucrose into a mixture of sucrose, fructose, and glucose. Sucrose is not a reducing sugar, and here is its structure. 4. Polysaccharides. Poly means many repeating units. An example of a polysaccharide is cellulose. It is a polysaccharide containing 7,000 to 12,000 glucose units connected through beta-glycosidic bonds. Cellulose strands strongly H-bond to one another, giving wood and plant material their rigid structure. Starch. Starch is also comprised of glucose units connected by glycosidic linkages. 2 components, amylose 20% and amylopectin 80%. Amylopectin has glycosidic branches of C6. And here are both of those structures. 5. Amino sugars. Amino sugars like glucosamine are important biomolecules. Acylated glucosamine forms an important polysaccharide called chitin. The N-acyl group allows for even stronger H bonding between the neighboring chains in cellulose, and oftentimes these are the exoskeletons of insects and arthropods. And glycosides. And glycosides are formed by reaction with an amine under acidic conditions. The nitrogenous bases are going to be the base pairs in DNA that are connected to deoxyribose units via N-glycosidic linkages, forming a nucleoside. Nucleic acids are the genetic code. Ribose forms ribonucleosides and ribonucleic acid RNA. Deoxyribose forms the deoxyribonucleosides and deoxyribonucleic acid or DNA. DNA. There are 4 different heterocyclic amines that attach to deoxyribose via N-glucosidic linkages to form the DNA. There are 4 different heterocyclic amines that attach to deoxyribose via N-glucosidic linkages to form the DNA nucleosides. In DNA, the nucleosides are attached to phosphate groups, forming nucleotides. The phosphate groups of the nucleotides are connected to make the DNA strand or the polynucleotide. H bonding. The nucleotides in DNA can attract one another through H bonding of the DNA base pairs. RNA. It's structurally different from DNA. The key components here are that the sugar in RNA is ribose. RNA contains a different base, and RNA translates the information stored in DNA into working molecules, proteins, and enzymes. 6. Review of key reactions. Hemiacetal formation, pyranose and furanose rings. Monosaccharide reactions. We saw acylation, alkylation, glycoside formation, reduction, oxidation to aldonic acid, and oxidation to alderic acid. Base chain length. We also learned that the length of the chain may be increased or decreased depending on the reagents used.